Okay, so hello again, organismal biology fans, and this video is going to pick up right where we left off. I kind of left you hanging, wondering what was going to be up with the oxygen in the atmosphere, and we're going to pick up there. So in our last episode, we started our road trip across the U.S. We're at a time scale here where 1 millimeter is 1 year, and that means that it's going to be about 4,500 kilometers is going to equal the 4.5 billion year history of the Earth. All of human civilization is right here on the beach, somewhere in Maryland. And we're going to start over here on the beach in California. And so what we saw last time was that the first about 700 million years over to almost the Nevada-Utah border, it's going to be about 7 hours of driving time at highway speed, if we don't stop for any breaks, is going to be what's called the Hadean epoch. We don't have much preserved record from that time. And because the rocks have been either destroyed or pulverized down to little bits, we don't have fossils. We do have some chemical evidence of life back there. At somewhere around 3.8 billion years, we get into what we call the Archean epoch. This is where we're getting large areas of preserved rock, what we call cratons. And in those cratons, we have fossils pretty early on. Again, there's chemical evidence of life going farther back, but we find good fossils that I showed you last time, starting at about 3.5 billion years. About in Utah, we've been driving for about 11 hours at highway speed to get there so far. And remember, we drive through all of human civilization in about 1/6 of a second at that scale. So we have life by about 3.4 billion years. For the rest of the Archean epoch, though, which is going to go for about another 900 million years, we only have prokaryotic life. And we have various reasons for believing that the life was only prokaryotic at that time. One reason for thinking the life was all prokaryotic at the time is if we look at the fossils, they're all tiny. So you can see, for example, these little bacterial guys that are in different stages of division and so on, but they're all very small, only a few microns. That's a 10 micron scale. They're in the size range of modern prokaryotes. They also, we know that by that time we would have had the features that we see in all life, including the prokaryotes, but we don't yet have the features that only occur in the eukaryotes like ourselves. We do find prokaryotes that are colonies, like these little chains. Again, their size is itty-bitty, bacterial-sized. We find larger colonies that we call stromatolites, like the ones we still see around today at places like Shark Bay. But it's not until what we call the Proterozoic epoch on our chart, that starts about 2.5 billion years, that we get two very important things and very closely related things happening. Number one, we're going to get eukaryotes, and number two, we're going to get oxygen. And I mentioned last time, and we'll look at this a little bit more in the next topic when we look at the organisms more. Having aerobic respiration and, if you photosynthesize, having oxygen producing photosynthesis are features that define eukaryotes. You can't, in particular, have eukaryotes that have aerobic respiration until you have oxygen, and you're not going to have oxygen until you have oxygen producing photosynthesis. So, actually, right around Scottsbluff, Nebraska, which has a really cool national monument to hike at and really good Mexican food, is right here in kind of the panhandle of Nebraska. 2.5 billion years ago, we've been driving now for 20 hours at highway speed. We finally start to see oxygen appearing in the atmosphere. And that's what defines the end of the Archean epoch and the beginning of the Proterozoic epoch. How do we know there's oxygen in the atmosphere appearing at this point? And how do we know there wasn't oxygen before, is the first question. Well, if we look at the geology of the time, remember that the continents are growing. We have this Archean craton here, for example, with North America, just the few northern states of the U.S., and then a good part of Canada. And then as we get into the Proterozoic, when we get up to about 2 billion years ago, and 1.7 and 1.6, we actually have a series of continental collisions that add a significant amount of crust down here into the U.S. So these are areas that are early Proterozoic up here, middle Proterozoic down here. And so what do we find if we look at rocks of that age in these parts of the U.S. and similar, areas of similar age in other parts of the world? Well, these turn out to be very economically important areas, particularly in the U.S. This strip up here that goes through sort of Minnesota with and Wisconsin is the major iron-producing area of the U.S. We found what are called banded iron formations. This is a map of Minnesota, and it's showing here these various strips of iron-producing areas that are not only for the U.S., the main source of iron ore, but actually one of the major iron-producing areas in the world. You have what's called the Siwalik Iron Range, in particular right here. This is a little description from a Minnesota geology website, and check out the age. This is a sample of banded iron from approximately 2.5 billion years ago, in other words, the very beginning of the Archean. What's banded iron? Well, one thing you can tell is it looks red. And you might be aware that one thing that happens with iron is that it turns red because it reacts with oxygen in the atmosphere and rusts and forms iron oxide. To have rust, the mineral here is called hematite. It's actually called heme because it's the same root as hemoglobin, which has red-colored, iron molecules. If you have rust, hematite, that means the iron must have been reacting with oxygen, which means you have oxygen that's in the atmosphere that's also able to dissolve in the oceans. And we don't find that until about 2.5 billion years ago. The reason it's called banded is that you find layers that are very high proportion of almost pure iron oxide in between layers that would have been more sort of typical mud. What's happening is that it's thought that what was going on was that you had iron dissolved in the oceans. For example, underwater volcanoes, deep ocean vents and so on, would release reduced iron into the water. And reduced iron is actually reasonably soluble in water. And so the oceans up to that time had a pretty high iron content. Once you get oxygen present in the atmosphere dissolving in the oceans, reacting with that iron, oxidized iron is very insoluble, and it would precipitate out. And so it's thought that what these bands correspond to were periods where there was an increase in atmospheric oxygen, like a little burst of oxygen, dissolves in the ocean, reacts with the iron, and precipitates out. And this happened over and over again to build hundreds of meter thick deposits of iron oxide. We find this type of rock for hundreds of millions of years in the Proterozoic. Eventually we don't find so much, and apparently what's happened is that the iron that was in the oceans essentially all precipitated out by reacting with the atmospheric oxygen. And so these early Proterozoic rocks then, formed when oxygen appeared in the atmosphere, are also economically the main source of the world's iron oxide. And many wars and such have been fought over iron deposits, all the way from prehistoric people, to the 20th century. And that's why they are where they are. So on our handout, when we get to the Proterozoic epoch, starting about 2.5 billion years ago, the continents are continuing to grow and drift and collide. And so we build up around that area of the continents, rocks of that age. In those, we find iron oxide, banded iron deposits, and that tells us that that's when oxygen was appearing in the atmosphere. We actually have various geochemical ways of estimating what the oxygen concentration was. For a long time, it was a lot lower than it was today, probably less than 1%, but it was there. And so we have this increase in atmospheric oxygen starting about 2.5 billion years ago. Shortly after that, early in the Proterozoic epoch, we begin to find eukaryotic fossils. And that's no accident, because one feature that all eukaryotes have is aerobic respiration. They're dependent on oxygen to metabolize their food. We find eukaryotic fossils going back about 2.1 billion years, a few hundred million years after oxygen begins to appear. And then finally, after about 2 billion years of aerobic respiration and oxygen-producing photosynthesis and eukaryote evolution, we finally find the multicellular organisms we're focusing on this course appearing right at the end of the Proterozoic. We get the oldest animal fossils. And that's in fact what defines the end of the Proterozoic. The period after that, starting about 600 million years ago, we call the Phanerozoic epoch. That's the entire history of animals, plants, fungi, multicellular eukaryotes happens within that. And of course, the continents are continuing to drift and collide and evolve themselves during that time. So on our time scale, starting after about 20 hours of driving, over at the western tip of Nebraska, we start to see banded iron formations. And somewhere towards central Nebraska, we begin to find eukaryotic fossils. The banded iron formations, again, iron oxide, particularly up in Minnesota, is the main iron-producing area in the U.S. Huge deposits, also other areas of the world of that age. Once there's oxygen in the atmosphere, we have the potential to have eukaryotic life. And we'll talk about eukaryotes more in the next video. But hopefully you're aware that one thing that eukaryotes have are these things called mitochondria, the sites of aerobic respiration. That's their main source of ATP. And being aerobic, they're dependent on oxygen. So you can't have eukaryotic cells with mitochondria until you've got oxygen for the mitochondria to use. They could be there. There's the potential because of the oxygen, but how do we know when we actually have eukaryotes? Well, the first cue is simply the size of the fossils. Here are these prokaryote fossils over 3 billion years old back in the Archean that I showed you before. The little spheres and ovals that are, you can see them at different stages of division, but they're all less than 10 microns in diameter, just a couple of microns across. When we get a few hundred million years into the Proterozoic, we begin to see fossils like these, Proterozoic eukaryotes. And one reason we think that all these things are eukaryotes is really simple. They're big. Okay, here are our little prokaryotic dots from the Archean by comparison. That scale's 10 microns. Here's a 50 micron scale for our eukaryotic fossils. And you can see that we get fossils in the Proterozoic that appear to be single cells. But those cells can be tens or even hundreds of microns across. And that's the size range of modern eukaryotes. In fact, just from size alone, you can tell very reliably if you're looking at a prokaryote or eukaryote, simply because mitochondria are endosymbiotic bacteria. They are prokaryotes living within a eukaryote host. The mitochondria are in the size range of prokaryotes. This much larger host eukaryote then has to be big enough to contain a number of mitochondria. So just this difference in size is a really good indicator that these guys must be eukaryotes. You don't find prokaryotes in that size range. You also begin to find, even in the early Proterozoic, starting about 2 billion years or so old, what appear to be multicellular, probably eukaryotes. This is one of the earliest here, a thing called Grippania, that goes back as far as about 2 billion years. Not real exciting to look at, but what you've got here is a flattened thing that appears to have some kind of a supporting structure. It's tempting to call it a vein, although it wouldn't be that because it's clearly not a plant yet. But it seems to have some kind of organization that maybe has the beginnings of specialized parts. At the very least, this is a large colonial organism. The cells of these are eukaryotic size. Very likely you're beginning to have some differentiation of cell types here, pattern formation, differential gene expression. So you're getting multicellular eukaryotes, probably multicellular algae, somewhat like the multicellular green and red and brown algae that you're going to see in the lab. But for reasons that we'll see, multicellular algae are not plants, and in fact most of them evolved their multicellular bodies independently of plants. It's not until the very end of the Proterozoic, about 600 million years, that we begin to see specimens that are, number one, definitely multicellular, but also almost certainly related to living animals rather than some separate origin of multicellularity. And it's not until somewhat after that that we're going to get actual multicellular plants. Fungi do not preserve well, terrible fossil record, but we do see some fossil record of fungi around the time that we see animals and plants moving on to land, kind of over here by the Pennsylvania border on our time scale. So back to our handout then. In the Proterozoic epoch, we have the appearance of oxygen in the atmosphere. That tells us, number one, that oxygen-producing photosynthesis has evolved by that time. But we do have photosynthetic prokaryotes, cyanobacteria, that have oxygen-producing photosynthesis. So probably that shows us the origin of cyanobacteria, which are still prokaryotes. But once there's oxygen in the atmosphere, we have the potential for aerobic respiration. And after a few 100 million years, we begin to find these larger fossils that, based on their size, must be eukaryotes. And if they're eukaryotes, they almost certainly had aerobic respiration requiring oxygen. Like I showed you, there are some probably multicellular algae that we find through the Proterozoic, mostly just colonial at best or unicellular. It's not until the very end of that we find multicellular animals. And that marks the end of the Proterozoic epoch and the beginning of what we call the Phanerozoic. And then the entire history of the living animals, the living plants, at least the fossil record of the fungi, is going to be in the Phanerozoic epoch, 600 billion years in the present. And that's only about 13-14% of Earth history for about the first 85% plus of world history. We have life. We eventually have eukaryotes, but we don't have animals or plants. So the diversity we see today, the world we see today, is a really different thing from what we had for most of Earth history. Scrolling down here then finally on our key points. Banded iron deposits, iron oxides, are evidence of atmospheric oxygen. It starts to appear about 2,500 million years ago. I'm going to move this around a bit. Sorry. There we go. Okay. Get myself out of the way a little bit too. Those banded iron deposits are evidence of atmospheric oxygen starting about 2.5 billion years ago, 2,500 million. That almost certainly marks the origin of oxygen-producing photosynthesis, which means we have cyanobacteria at that point. We get larger eukaryotes appearing during the Proterozoic once there's enough oxygen for aerobic respiration. but the whole fossil record of animals, plants, fungi is in the Phanerozoic epoch, and that's the last 15%. So that's our introduction to the most of Earth history. And what I will do on the next video then is talk a little bit about the diversity of the organisms that we had back then, but also use that as an opportunity to introduce you to some ideas about what we call phylogeny, which means reconstructing the tree of life, the relationships of organisms, by taking a little bit of a look at the phylogeny, the evolutionary history, the diversity of those early prokaryotes and eukaryotes. And then from there, we're going to go into the plants for several weeks. And Dr. Griffith will take over the videos at that point and march you through the plant diversity and some of their physiology. I'll pick up again with the animal things a few weeks down the road.